Welcome back again. So in the last video, we talked about how the loss of evolutionary potential is one big negative effect of low genetic diversity, and inbreeding is another negative effect, which we'll talk about here. So inbreeding is most likely to occur in small populations, and the result is increased homozygosity. So what I'm showing here is one example of a species that has been affected by inbreeding, the elephant seal that was hunted to near extinction. They had really small populations with really low genetic diversity. It led to a lot of inbreeding or mating between siblings, and it led to effects like spinal deformities, which are shown here on the right. So overall, inbreeding, we said it increases homozygosity. It also leads to alleles that are called identical by descent, or IBD. So let's focus here on this fish pedigree, on the recessive allele. So one quote-unquote grandparent fish has this recessive allele. They pass it on to two of their children fish. So we have two heterozygotes here. What happens is if you have inbreeding where two full siblings are mating, they can end up producing a grandbaby fish, for lack of a better term, that is homozygous recessive. So this individual is inbred because it's the result of mating of two full siblings, and it has these two recessive alleles that are identical by descent because it has those alleles through its descent, through its lineage, as a result of originally coming from this one grandparent and then being passed down to it. So when we're thinking about how do you quantify inbreeding, you can use a couple different metrics, which we'll talk about in this video. One of them is called an inbreeding coefficient, abbreviated as F. And like we've done previously, I'm not going to ask you to know how to calculate this or anything like that, but just know in terms of interpretation, if you see an F value of 0, that means there's no inbreeding, so the population has high genetic diversity. And if the F inbreeding coefficient is 1, that means there's complete inbreeding, essentially that individuals are genetically identical. So again, you don't need to know how to calculate that, but just that's how you would interpret it. 0 is no inbreeding, F equals 1 is complete inbreeding. So if we think about what types of molecular markers are used to do these calculations, some markers that were used more predominantly in the past are microsatellites, which remember are these repeats of different nucleotides, and now SNPs are also commonly used, which are more readily available, of course, because of the increasing availability of high-throughput sequencing techniques. And so if we do have a genome sequence available, one of the things you can do in terms of using molecular data to calculate inbreeding is you can actually calculate not only that inbreeding coefficient I just talked about, but you can also use genome-wide sequencing data to identify what are called runs of homozygosity, abbreviated as ROH, to calculate another type of inbreeding coefficient called F sub ROH. So this particular inbreeding coefficient is specifically based on these runs of homozygosity. So what the heck does that mean? There's a bunch of going on in this graph, but I'll walk us through it. So this is an example from a recent paper. They were looking at inbred populations of the Isle Royale wolf. And so you might notice here this wolf also has a spinal deformity. A lot of individuals in this population that are inbred have this spinal deformity as a result of inbreeding. So that population is shown here in F. So what we're looking at in each of these panels, on the x-axis is the chromosome. So remember, in order to do this type of inbreeding coefficient calculation, you need a whole sequence. So you need to know where these different markers are in terms of where they are on the chromosomes. So that's the x-axis. The y-axis is heterozygosity. So the higher the heterozygosity is, the more genetic diversity there is, the higher the bars are on this graph. And so if we look at these different populations, so each of these different panels, A, B, C, D, E, and F, are based on different populations of wolves. So they represent different populations of wolves that we also know have different levels of inbreeding. So this Ethiopia population, for instance, we know is severely inbred. And if you look at its levels of heterozygosity, except for this one region here, it's all super, super low, especially compared to populations like in China where they're more outbred and they have much higher heterozygosity. So in Ethiopia here, this is what we would identify as a run of homozygosity, where essentially the entire genome is a run of homozygosity. And so the longer the run of homozygosity is on these graphs, the more inbreeding we can infer and the higher the inbreeding coefficient would be. So that's how you interpret these types of data, and that's how you use runs of homozygosity to identify whether inbreeding is happening. Inbreeding leads to high homozygosity, which means to these long stretches across chromosomes where the heterozygosity is basically zero. So if we think about inbreeding extending now to fitness consequences, inbreeding threatens the survival of small populations, specifically when it leads to these reductions in fitness. And if that inbreeding leads to these corresponding reductions in fitness, that's when we say we have this inbreeding depression. So this is some graphs, some data from a study in 2010 where they were looking at, a Swedish metapopulation of southern dunlins, which is a type of bird, a migratory shorebird. Unfortunately, this picture here was a little bird from within an egg that actually didn't hatch, and it didn't hatch because of this inbreeding depression. So these two graphs on the right here are showing correlations between several different metrics and the relatedness of the parents. So band sharing is the way they were quantifying relatedness. 0.5 here on the far right hand side is higher relatedness of the parents. And if we look at several different fitness metrics in terms of the total hatching success or the proportion of heterozygous loci in the chicks, there's negative correlations for both of those. So both of these are metrics of fitness. For both of them, the more related the parents are, the lower the fitness is. So this is evidence of inbreeding depression because we actually have evidence of reductions in fitness. So let's think about how inbreeding leads to inbreeding depression itself. We'll talk about a couple of different ways this happens. The first mechanism is known as dominance. So overall, the increased homozygosity that results from inbreeding means that deleterious or harmful alleles are more likely to occur as homozygotes. And therefore, when that happens, they can't be masked by a dominant allele. So an example of this is shown here on the right. This particular bird in this population, they are suffering from a type of lethal blindness. If you look, its eye looks a little funky. So overall, say that the recessive allele lowercase b is what causes this blindness. If you have a heterozygote, you have a one dominant allele that can mask it, so that blindness characteristic is not going to be expressed. But if you end up with inbreeding, where now you have a loss of heterozygosity, and so now you have high homozygosity, and you have individuals that are homozygous recessive, that means that lethal blindness characteristic is going to be expressed. That can lead to this reduction in fitness and inbreeding depression. Another mechanism that can lead to inbreeding depression is what's called overdominance. Overdominance can lead to inbreeding depression because you have these advantageous, beneficial heterozygotes that are lost following inbreeding. Again, the overall result of inbreeding is an increase in recessive homozygotes. So the classic example of heterozygote advantage and overdominance is sickle cell anemia, where if you are a heterozygote, you are resistant to malaria. If you have two copies of that recessive allele, you actually have sickle cell disease. So in inbreeding, what happens is you lose these heterozygotes. So you lose the benefits of having that one dominant allele and one recessive allele. And now all you have are 2 copies of the recessive allele. So that can lead to reductions in fitness. In this example, everyone would now have sickle cell anemia. Another really interesting way that inbreeding depression can result is from changes in gene expression. And they did this really cool study, which is from 2005 now, but it's still very striking, I think, in the result in terms of what they found is that if you were an inbred individual, and they were doing this in Drosophila, an individual that is inbred and in normal, non-stressful environment actually shows gene expression that indicates they're stressed. So they have increased gene expression of things related to metabolism and stress, like heat stress. So overall, if you're an inbred individual, your gene expression is acting as if you are in a stressful environment because inbreeding itself actually acts as a stressor, which I think is super cool. You can actually see that at the gene expression level. And what's also really interesting is, so that's what happens under normal conditions. If you then put an inbred population into a stressful environment, that gene expression difference and that difference in fitness is even more pronounced when you compare an inbred population to an outbred population. So here in this graph, the outbred population is in blue, the inbred population is in red. So under these normal conditions, the inbred population already has a reduction in fitness. But when you also put that inbred population into a surrounding stressful environment, that difference becomes even larger because now you have this interaction. So when you're put in a stressful environment, that difference in terms of gene expression, where the inbred populations are upregulating those stress response genes, that difference is even larger in a stressful environment. One thing I also learned while I was reading this chapter is that inbreeding itself can also directly interfere with stress response pathways like the heat shock protein expression. So in this graph, the y-axis is HSP expression. And we have different populations. The white bar is an outbred population, and then we have 2 different levels of inbreeding. The x-axis is showing the response of HSP in different temperatures. So normally, under high temperatures, we would expect HSP expression to increase. That's part of the protective response for the organism. We can see that this does happen for the outbred individual, but the inbred individuals actually cannot increase their expression of HSP as much. So this provides evidence that inbreeding itself can actually directly interfere with stress responses, which is also very interesting, I think. So that's bad if you have inbreeding interfering with stress response, but there are also some processes that can reduce the effects of inbreeding, which we will talk about next. So overall, inbreeding increases the likelihood that bad traits, harmful traits will be expressed. What can happen though is if you expose those traits, so let's think about those population of birds where they were all blind and they all ended up dying, right? If you're a blind bird that dies, that means you can't reproduce. So any individuals that are able to survive would be the ones without those deleterious alleles. So this idea of purging refers to the fact that once you have those exposed traits that they're selected against, those deleterious alleles can be eliminated or purged from the population. So the effect would look something like this. In this graph here, we have generations of inbreeding on the x-axis and relative fitness on the y-axis. So overall, once inbreeding starts, we see a reduction in fitness. But if purging happens, we start to see an increase again in fitness because what's happening at this inflection point here is basically all of the individuals with those lethal recessive mutations are dying. And so any individual that is left that is able to reproduce has the quote-unquote good genes, which can then increase fitness. It is important to know there's not a whole lot of evidence for this, but it is one potential mechanism, and there are some examples from lab situations. Another thing that can happen is balancing selection can lead to high levels of diversity at some specific genes that have a functional benefit, even if the population is super inbred. So this picture here on the left is a Channel Islands fox. This species, these populations, have historically, classically low genetic diversity. So in one particular island, I think it's San Nicolas, the individuals there have basically no genetic diversity whatsoever. But what's interesting is that at some particular genes, like these MHC genes that are involved in immune response, they actually have high levels of diversity there that are maintained by balancing selection. So overall, even if inbreeding is super high across the genome, if you have some diversity in these functionally important genes, it can still help the population to survive and persist. All right, so if we want to think about how to measure and infer inbreeding depression, one of the things that you can do is something called a heterozygosity fitness correlation, or HFC. Excuse me, letters are hard. So an HFC can infer inbreeding depression by identifying a relationship between low heterozygosity and reduced fitness. So essentially what you're doing here is saying that if there's low heterozygosity, there's also reduced fitness. That is evidence for inbreeding depression. So this graph here on the right is showing that overall, the higher homozygosity there is, the lower fitness is. In this case, that fitness is represented by reproductive success, so number of chicks. So there's a negative correlation here. That's a heterozygosity fitness correlation that indicates inbreeding depression is indeed happening. Another really important point to make here is that the type of marker you use to calculate these HFCs is really important. So in this study here, they were looking at different populations of harbor seals. And what they found is that they got different results depending on whether they used microsatellites or SNPs. So this graph is a little bit complicated, but the overall idea here is that if we focus on the p-value difference when you're using microsats versus SNPs, the p-value when they used microsats was 0.06. So basically the correlation between heterozygosity and fitness was not significant, but when they used roughly 1,500 SNPs, they found the significance value was much smaller and was significant. So it's important to think about what type of markers we're using, how many markers we're using. Sometimes using more SNPs can lead to more accurate calculations and therefore more accurate estimates of whether inbreeding depression is happening.